Okay, hi everyone. I hope I will interest all data scientists and people who like to do lots of computational stuff. Because when I was going to be a neuroscientist, I didn't know that I'm going to do it. And I am biophysicist, and I have very, very little programming background. But when you get into neuroscience and you want to be a good neuroscientist who combines lots of fields together, you have to do lots of programming and data science stuff in your neuroscientific career. So today I am going to cover, this is the outline of my talk, so neuroinformatics, so I'll just tell what it is. Then I will talk about neural features. I want you to get to know some of the terminologies that I'm going to use during my talk, because that will really help you to understand later what I'm going to talk about. Then I will show and talk a lot, or partially about the eye, because that's my favorite neuroscientific part of the body. And then we have neural plasticity, where I will present actually my research that I have done in UC Santa Cruz in Stanford. It was a combined project. And of course, then I will conclude what I was talking about. So I want to get to know my auditory as well. So please raise your hands if you are a physicist. Okay, nice. A programmer, data scientist, wow, that's a lot. And we have physicists, we are as well. Data scientists, that's nice. How many of you are biologists? Oh, that's cool. Okay, I'm not alone then. Then who has a physiology or a neuroscience background? None of you, okay. How many of you have in like at school or wherever, the neuroscience class? Okay, yeah, I don't know if you remember or not, but I remember that I was struggling a lot for my neuroscience class at the school because we had only one lesson and it was covering everything and it was so messy that I wasn't really sure why I like it so much. In any case, so I can see that lots of you are mostly programmers, so that makes my presentation nicer because I prepared it for you guys. So let's move forward. Okay, so before we actually go forward, I want you to know that we have, as in a computational world, the very small component, which makes basically everything. In the nervous system, it's basically a neuron. A neuron is the cell which is responsible for all of the signaling and information transfer in the human body. And I have, for the physicists, maybe you will get this better, but I'll try to make it much easier. So let's say we have an electrical circuit, which is, let's say, a microphone. Yes, I'm holding a microphone. It's my input device. Then we have a control system, which is an amplifier, which makes my voice higher. Yes. And then we have an output, which is the loudspeaker. That's why you can hear me better. Yes. So the same thing is a neuron. So neuron does have an input. It's called dendrites. And it's kind of the same thing that a microphone, because it receives input. Then we have a controller, which are the axons. And the axons are very important because they are speeding up the information transfer. And I can go deeper and tell about the myelin, but I will not go. So just keep in your mind that the axons will make very huge difference into the signal transferring. Then we have an output, which is the part where the cell is basically giving the signal that it received, amplified, or changed as well. And the output is basically connected to the different cells, neurons, and they are seeing different things as well. So I just want to show this is the schematics, and this is a neuron in a brain, which requires tons of work to get this picture, because when you are looking into the cells, into the brain, you do apply lots of colors and lots of steps in order to make them visually much more nicer. But then in order to get the 3D reconstruction, you have to do lots of programming as well. And I'll talk about it a bit later. So in general, I don't want to scare you, but this picture looks quite messy, and you can see lots of blue spots that are the cells, and lots of different colors, which are different cell types. And they are this is very, very small portion of the brain, and very, very few cells that are in there. So now I want you guys to do with me some math. Let's count how many interactions the neurons will have. So in our brain, actually, we have 86 billion neurons. We have a single neuron which makes 1,000 connections. So the output is 1,000. So just make your calculations in your brain. At the same time, we have about 22 billion glia cells. The glia cells are very under-discovered cells in the brain, but I did my PhD as well on this topic, and I can tell that actually glia cells can control lots of stuff, and especially the signal transduction part, the axons. One of the glia cells is very, very important for that. So, which means that it is adding another component into the signal transduction. So you can imagine how complex it is and how many different type of connections and networks we will have. And of course, I wanted to multiply by 3 because we have different features: the morphological, functional, and genetic. And I don't want you guys to be confused about the features. So that's why I kind of made a very, okay, in the other slide, I'll talk about it, but I'll get back to that. So what I want you to as well keep in your mind that neuroinformatics is very new scientific direction. It's actually it's about 20 years old, or if not less, because it has been created by the US National Health Institute, as we had so much data to deal with that they had to compute the data that we have collected, all the researchers, and try to make sense out of it. Because we have in the world so many neuroscientists, and most of them do kind of similar type of work, so we want kind of to bring all of them to one point, make a goal, just one, and work together. So basically, if you are a data scientist, you can actually do in the neuroinformatics like data analysis, which is not just going and taking the data. You have to really simulate the data. Like when you do recording, especially in the neuro system, you have so much noise, you have to clear that noise much sufficiently, and it's still a problem. And then you can do image processing, which is whole another world. And then you have a modeling of the nervous system, just to deal with a whole bunch of data that is there. And of course, the prosthesis. And the prosthesis is not only prosthesis for the people who are impaired, like they don't have legs or like arms, and you are making them, but as well to make eye prosthesis. Which is very hard, and I am kind of part of that project, which I don't know when it will end. Yeah, it has been established late 80s. Okay. Okay, before when people were doing classification or were trying to concentrate on the neurons, first it was just the morphology. They were looking into the morphology, and what was very interesting that before even electron microscope was discovered, electron microscope is a method, microscopic method to look into the cells where you can actually see very tiny nanometer like size, like proteins and everything. But people weren't able to do that. They just had a light microscope, some lenses, and they were just looking into the tissues and trying to see what are there, and they discovered that there are so many different types of neurons. The physiology part came a bit later because the physiology was dealing more with a physical component of it. As you have seen in my second slide, I was trying to make that point that neuron is actually electrical circuit. So it's basically is a physical living organism in our body. And of course, the molecular component, I think in our previous talk you have heard about it quite a lot. It's basically genetics of the cells. And all different cells have different genetic profiles, different morphology, and different physiology. They have some common things together, but they are really, if you take into consideration all of them, then the picture becomes even more complicated. And that's what now we are doing. That's what nowadays classification, neural classification looks like. And we have a joke in our field that each neuron is like an individual, like we all are, and they are all different, although they can share some of the features with each other. So the morphological profile, how we get there. So we have a mouse. That's the basically animal model that I mostly worked with, although I worked with lots of other animals. But anyways, you take the tissue, because I'm working with the retina, I show you the retina image, and then you are, then you are isolating the retina, and then putting lots of different fluorescence dyes, because you want that you see different cells. So that's why each cell you find a protein which is specific for that cell, and then you stain for that specific protein, and you basically end up getting a picture similar like here. Then you zoom in, and then you look into the cells, and depending on the task that you have, you can go to do the 3D reconstruction or just look into the different pattern of the cell clusters. So here we have the functional profile. You see similar thing. You are removing the retina, but instead of fixing it in order to get a nice image under the microscope, you actually record from real tissue. So you basically take the tissue, put on the microelectrode array. So you can see here, I know my slides are a bit messed up, but that's okay. So we have here, I don't know, my pointer. Yeah, that's a microelectrode array. And this basically array of the electrodes where you basically do record the signals that come from the cells. Because when you show the cells, let's say the visual signal, black and white, I don't know, screen, the cells do see that, and they basically send a response to the brain, okay, I see black and white squares. And that is recorded on this electrode. Wherever cells is, you will see in the output. Okay, that's the different stimulus that we are showing, and that's the neurons that you will get. So you will basically get physiological profile of the cells. And usually, nowadays, the best thing would be to combine, let's say you have the physiology, then you are fixing your slide, and then you are basically looking into the morphology, and then you align the physiology and morphology, and you are saying, okay, these cells are morphologically similar, but physiologically they are completely different. And of course, the extraction of the RNA and gene extraction, and you get the extracted gene from individual cells and the genetic profile, which means that each cell does have a selected amount of proteins that the other cells doesn't have, and that makes them very, very different. And whatever I have shown you here is done from the one single retina. So this is the data, and you can go, and if you are interested, you can go and check the paper that I am referring here. It's very interesting, and there is lots of competition, so you can get to know how the analyses have been done and the combination of different data sets have been done. Okay, so now I want you guys to go and try to get into my world of the retina. So I have started working with the retina when I was doing my master's. And I did very, very simple project here at the Biochemistry Institute, where I was just making bright light into the retina and was trying to see how the cells are damaged and then comparing with the healthy one. It was very fun and cool. But I didn't know that I will get into it when I was going to do my PhD, because when I started my PhD, originally I had to do some genetics related thing with the retina, but I ended up doing physiology. And that makes me to dig into the world of the retina. And in the retina field, we have another joke that we are telling that the brain is just an extinction of the retina, because the visual system is involved in so many different areas. It is involved in all the sensory systems, and it is enriching our perception of the world. So now let's kind of go and see where the retina degeneration stands. So globally, about 80% people do have some type of blindness, and this is basically some chart which is mimicking that. So age-related macular degeneration is one of them, and very many people do suffer with this. And with the research that I am trying to do, I'm trying to understand when you even fix the gene that is not working right, how you can make the retina to go back. Because as you know, nervous system is not really regenerating. We don't have really new cells appearing there. So that's why it's very important to see even if the cells that are there, they will change or do some kind of arrangement in order to fill the gap of the dead cells that are there. So there are basically 200 different gene mutations. There are more than 100 cell types which are involved in this, and all these cells make complex connections, and the retina is just not a one layer of cells. And here are some more details and some more numbers, how many people do suffer from that. But I just want to make sure that you understand that not all the people do suffer being blind in the end, because some of them, like for example, the glaucoma and so on, you can kind of fix that kind of blindnesses. So I'm talking about the genetic ones, like macular degeneration. So before we jump into the research that I have done, I want to make a point that not all animals don't regenerate. For example, the zebrafish does regenerate. They actually make a new cells, new neurons, even if they are dead. And I hope this video will play, or maybe it wouldn't. So let me see if I click on it, it will play. No, it doesn't. No. But I wanted to show here you how the cell formation, like how you can see just a couple of cells, but in the end of the video you would see just the one whole organism. That's the live imaging, 72 hours. The zebrafish was under the microscope, and it was taken the image under the microscope for 72 hours. But unfortunately, I cannot play it. Okay, let's move forward. It's okay. So neurons can kind of recover. They regrow new processes. They as well are able to make new connections, and as well they rewire. And that's what I'm going to prove you in the end of my talk today. And here I have another video. Hopefully it will play. So how? So I have to go to the browser. Oh, okay. Full screen. Let's play.
Sense of sight allows us to orientate ourselves to what is around us. When we lose our ability to see, we lose our sense of orientation. A healthy eye functions like a camera. It focuses the incoming light rays and projects them onto the retina. Two types of light-sensitive receptors in the retina, the rods and the cones, transform these light patterns into electrical signals. The signals are then transmitted to the optical nerve, which relays the information to the lower surface of the brain. Here, the image is assembled. Many eye diseases can be treated with glasses, contact lenses, medication, or even. Retinitis pigmentosa is characterized by progressive dying of the light receptors in the retina. If the receptors begin to die starting from the periphery, the affected person only sees what is in front of him. If the receptors begin to die starting from the center outwards, the vision of the affected person becomes restricted to the periphery. Reduced vision, night blindness, and a restricted visual field are the main symptoms of this hereditary disease.
ankommt. Das sind welche Krankheiten, die Sie haben.
Yeah, I just wanted you to kind of get to know how. Presentation. Oh. Okay. Sorry. Yeah, so that you kind of get to know what retina is, just with interactive way, because I'm going to talk about it. Oops. So now we will move into the neuroplasticity part. And there are 4 different ways to kind of play around with this disease. One of them is optogenetics, and lots of bioinformaticians are actually involved in this research, where basically they are creating channels. As you remember from the video, there are the photoreceptors, the light-sensitive cells that you actually can see with them. They are dying. And when they die, you are left with the cells that they cannot see the light. So which means that you need to make them light sensitive. In order to do that, you have to insert some light sensitive protein. And luckily, we do have that kind of proteins in the nature, like a green algae. They do have the light sensitive proteins, which are channel rhodopsin channels, basically. And what we do, we are just basically modifying them. So you are taking them and playing around with the different components of the active zone of the proteins in order to make them to be opened faster or slower, or be activated with the different colors or with a different contrast. So it's basically a very, very wide range of the things. There are some limitations because the channel cannot be changed for ever. So there are some limitations that the channels can do or cannot. That's why we cannot just have like fast-acting channel and as well slow-acting channel. It can be only fast or only slow. So that's why this is not really giving us complete answer. The next thing is the electrical prosthesis, and in the world there are actually 3 different labs who do this. I was part of 2 labs, luckily. So one was in Germany, which I was doing PhD, and the second one is in Stanford, Santa Cruz, where they are basically developing actually prosthesis. And the idea of the prosthesis is basically you are having a chip inserted under the retina. Again, they are not light-sensitive cells. So the idea is that this chip can see the light, and it transfers the light signals into the electrical signals. And these signals are transferred to the cells that are already there. And then the brain kind of sees what is happening. With the electrical prosthesis, we cannot ever recover the color vision. So these patients see only shadows. So they can actually see things, they can see the trees, they can see the street, they can see the signs, but they can never see the colors. The next step is the gene therapy. So the gene therapy is basically you are trying to find the gene that is messed up and fix it. But the problem is that when the patients go already for this on the stage, it's already too far. So you cannot really anymore do much. You can stop it, but still the central part, let's say macular degeneration, where the central part is very essential because there are, you can see much sharper. With the peripheral part, it's very blurry. So basically your vision is not really good if you have only periphery. And the part that I was working, it was the basically healthy retina state when you are kind of trying to see, okay, you have fixed the genes. Now you ended up having the parts which don't see. So what it will happen with the brain, how it will deal with it. So that's what I was working with in Santa Cruz and Stanford. So I will drive through to the process that what we were doing. So if you remember from the video, that's the eye and that's the retina, basically. And you remember that's the light-sensitive cells. Then we have an inner part of the retina where lots of coding and information change is taking place. And then we have an output cells, which basically take the information and bring it to the brain, upper brain regions. And that's basically the layers that I was talking about. In the macular degeneration, you basically don't have the photoreceptors. So if the photoreceptors are gone, the bipolar cells are remaining, and they have to do something. So there are still some photoreceptors left, so they have to do something to connect to the photoreceptors that are left there. It's not working very well. Okay. Yeah, so that's what we have left. So now let's see how I am using the method to kind of eliminate part of the retina. So the diabetes retinopathy is one of the blindness-related diseases where basically you have too much energy, diabetic retina, and it is used to grow the blood vessels underneath the retina and basically makes the scar like kind of thin layer, and you cannot see it. And if the photoreceptors can see, they start slowly to die. That's why these patients have to go and get treated, which means to remove the blood vessels. But if you remove the blood vessels, that will not solve the problem. What is solving the problem? To create another part in the retina which will use the energy. So what is done, they are laser ablations, so you can see here that's the normal retina and this is ablated retina. In the peripheral part, you basically go with the laser and specifically burn photoreceptors. So which means that now retina has something else to work on instead of building the blood vessels. And this is the method that I have used. So basically what I have done with the mouse retina, sorry, my slides are a bit, because the format is different. So I have a mouse retina or rabbit retina, squirrel retina, because I was working with all of them. And I was placing them under the laser microscope and was trying to remove some parts of the retina. And that's how the schematically it would look like. So some part of the photoreceptors are gone. So we still have intact inner cells and we still have intact the output. And we are calling the part that the cells cannot see the blind spot. And depending how big the blind spot is in the retina, your brain would be able to see or not see it. If you have too big blind spot, of course you would see a blind spot into your eye. But the blind spot, if it is too small in the brain, you do have as well some overlapping of the circuits, so kind of brain tries to predict what is shown, and actually most of the things that we see visually, they are prediction as well. So we are kind of predicting the changes, our visual system predicts changes, and that's how you see the things. So the experiments that have been done in a rabbit, so I want you to kind of pay attention in some of the terminology here too. So rod bipolar cells would be the cells that I was concentrating on. Oops, I don't know why. Okay. So I was concentrating on them because they were the cells that they were still there. And then the pinkish ones are the connections, functional connections between them, because I'm going to show you histological slides so that you understand what the pink spots are. And then the laser came in, and then just in order to kind of recall what I was talking about, the morphological profile part, that's what happens. So basically you are staining the cells that are remaining, and you are having this picture here. So this is the schematic, and this is just the histology image, the morphological image. The green cells are the rod cells. These guys here, rod bipolar cells. And the red dots are the functional parts, and you can see in the middle we almost lost the functional part because the photoreceptors were gone, so which means that these cells that are in the middle, they are not going to see anything. And what is happening, that when you are ablating the blind spot, over time the cells do actually change, and here how they are changing. Over time, the spot is getting closed. So here is the ablation done in 14 days, and then after 30 days we are dissecting the retina, and we see that the blind spot is kind of getting smaller. After 60 days, it's quite similar to the 30 days, and the 120 days you can actually see in the middle, the bipolar cells look different. And this is done just a simple confocal microscope. There are no reconstructions or anything. But in order to actually understand what is happening, you have to go and do more analysis. And most of the time, the analysis that it's done, it's manual because you cannot find good apps or programs that would do it. So, okay. This is the functional part of it. So remember when I was telling that we were doing recordings from the cells? Yes, the microelectrode arrays. So what you see here is basically the same tissue here put on the electrodes, and the heat map shows with the red the parts of the retina which are responding better to the light stimulus, and the blue ones means that you don't have any response. So you can see that morphological and functional components do align with each other. And as well, over time, after 4 months, you would see that the blind spots really did get smaller. So we are showing that functionally, there is something happening. Doesn't really mean that if the photoreceptors are gone in the middle, that's it, that's that story. There is still something going on in the retina. The cells are getting rearranged and they are making connections with the neighboring photoreceptors. So that's basically the functional recovery of the retina over time. And that's the blind spots that I was talking about. So now let's see how healing looks like in general. So this is the actually retina from the mouse, and they are spots, not the lines, ablations. And you can see, here we have a normal bipolar cells, and we have restructured ones, which means that they have changed their structure. And I have, for simplicity, a couple of examples of them. So that's a normal bipolar cell. So what you see here is the cell, and purple dots are the functional parts. So they make lots of connections with photoreceptors. And this is the restructured one. So here I have an ablation area, and you can see that the bipolar cell, instead of having this kind of dendrites or branches, it kind of reaches out to the lesion edge and tries to make connections. This was such an interesting and cool discovery for us because we were like, okay, there are lots of experiments that are done that people were trying to implant photoreceptors and they were not working. But why they were not working, we still don't know. The only idea would be that if you try to have an embryonic tissue implanted into the already adult retina, the cells are having different expression of proteins. They are in a different stage, so that's why the integration is not working. But what we have shown with this, that if the correct photoreceptors really be implanted into the retina, they can get integrated, because bipolar cells, they want to make connections. Because if they don't make connections, they would die, and they don't want to die, apparently, because they make connections. Okay. So another thing is basically that's what I was trying to tell, that this cell basically lost all the connections, and you can see that it lost the processes, and in the end, which is on very, very in the middle, the cell, and cannot really reach out to the borders, it will basically die, and we will end up having blind spot in the very center. So what I want you to compare, this is the scar tissue. So if you have a wound, it takes really a while, yes, to have a scar recovered. So you can see that's the 4 months timeline, which I was trying to prove that the same thing is happening with the retina. So we do have scarring as well happening inside of the retina. And it takes time. It's not a very simple and easy process. So just to conclude, that's basically what I have discovered. So that you have a normal bipolar cell, and then when they lose the connections, they first try to go up, and then they are going anywhere just to try to find the connections, and then finally they reach out to somewhere that there are photoreceptors, and they are making connections. So that brings us to the point to conclude that retina is really plastic, that we have restructured cells which are making new connections, and as well that the blind spots get smaller over time. That's the slide. What I want to make a point in the end is that my research kind of makes us to the point that everything is not lost, and we can actually use the recovery function of the retina if we are using the rest of the things correctly to work properly. And if in order to get a full vision restoration, first we need to fix the genes that are not working well, and then find correct way to implant the photoreceptors as well administrate the correct proteins there. I haven't shown the part of my research on that because it would be too much into. If you are interested, I can talk about that with you. Just on the side. But yeah, we have looked into the different proteins and we tried to find out if they are actually playing a role into the connections, and we found a couple of them that have a key role into speeding up the connections or making the functional connections really proper between the cells. And yeah, this high sufficiency, normal and good photoreceptor integrations and fixing the problem on time, not waiting until the last minute. And all of you, I mean, I can see the audience is mostly young people, but when you get older, you have to kind of pay attention on the signs. Like if you see some spot that always follows you and it never goes away, that means that you have degeneration started into your retina. And if you wait too long, that will get much more deeper and much more many cells will die, and it will be much harder to recover them. Yeah, so I want to thank my lab, the Dr. Alexander Scher and Daniel Palanker. So Scher is from UCSC and Daniel Palanker is from the Stanford University. And thank you all for the attention. I guess you can ask questions if you have.
Okay, thank you for the event. I'm a journalist, and turned on. Well, thank you for the event. Thank you for the event. I'm a journalist, and I'm writing an article about the Barcamp and your speech, your talk. I want to know what do you think about traditional medicine? Can it prevent eye blindness, or what do you think about eye exercises?
Okay, so there are different things. So eye exercises would definitely improve your vision if you have mechanical problem. Like if your muscles are or the lenses are not properly functioning, that will help. Traditional medicine, it might help because the pharmacology or whatever we are trying to administrate into the retina to make it work better is basically proteins, and you eat proteins, and depending what you eat, that will make your vision better or worse. And what I was doing during my master's, actually, that was the point. We were looking into the taurine, which is basically very good antioxidant, and we were trying to make a point that if you have, when you are doing the surgery and you have taurine administration into the eye, then you basically have the recovery process faster. So of course, that can be the case, but first of all, we need to find correct proteins that are having influence, and then maybe make a suggestion, changing a diet or something like that.
Okay, thank you very much.
Հա։ Ուղղակի իմ գիտության հետ կապված ես անգլերեն եմ ավելի հեշտ է ինձ անգլերեն խոսալ։
Ուզում եմ իմանալ չորությունը աչքերի կախված է,
Չէ, դա չորությունը կախված չի դրա հետ, որովհետև դա ցանցատաղանթի հետ կապ չունի։ Ցանցատաղանթը դա ոնց որ ուղեղի հատվածն է, որը գտնվում է աչքի տակ, այդ որ դուք ասում եք, դա շատ կոմպյուտերին նայելուց, կամ քիչ քնելուց, կամ լավ չսնվելուց, ուղղակի սնուցումը աչքի վատանում է, դրա համար չորությունը առաջանում։ Տարբերություն չկա։
Neuroinformatics-ի ինֆորմատիկայի մասից մի երկու բան կարող եք ասել, օրինակ, ինչ pipeline եք օգտագործել այդ դատան, որ process եք արել։
Վաղը որ գաք, սա այդ մասին խոսալու է, բայց կարամ կարճ ասեմ։ Կախված ինչ ենք անում։ So for image processing-ի համար հիմնականում օգտագործում ենք MATLAB, մաթեմատիկա։ Այդ ծրագրերը կան որոշ app-եր, որ օրինակ Fijin կամ կամպանիաներ կան, օրինակ Size-ը կամ Leica-ն, որոնք որ իրանք իրանց confocal միկրոսկոպներն են ստեղծում, նաև փորձում են visual analysis-ի պլատֆորմեր ստեղծեն ու ծախեն, բայց ոչ մեկ perfect չի անում։ So իմ անալիզների մեծ մասը ես եմ արել, ծրագրավորել եմ, կարելի ասել, այդպես լավ ծրագրավորող չեմ, բայց դե ման ես գալիս գտնում ես ձևեր։ Ու MATLAB եմ օգտագործել։ Բայց եթե օրինակի համար ֆիզիոլոգիական data-ի հետ գործ ունեք, հիմնականում հիմա Python են օգտագործում։ Չնայած MATLAB-ն էլ է շատ օգտագործում, ես MATLAB եմ կոնկրետ օգտագործել։ Դեղ կա նաև Igor Pro, էլի նորից էլի ֆիզիոլոգիական տենց կամպանիաներ կան, որ սարքեր են ստեղծում, իրանք առաջարկում են տարբեր անալիզներ, բայց էլի հարցենա, որ մի պահ է գալիս, քո data-ն NZV feature-ները ունենում, որ դու պետք է ինքդ implement անես անալիզները, էլի։
Այսինքն վաղը workshop-ի ժամանակ, ինչ որ ձեր research data-ի վրա workshop եք անելու, ես ճիշտ եմ։ Մտածում եմ տենց բան անեմ, բայց հասկացա, որ ավելի լավ է մի հատ introductory բան ստեղծեմ, որ բոլորը հասկանան, թե data-ն որտեղից է գալի ու ուր է գնում։ Դա արդեն մի քիչ ավելի խոր բան է, որ data-ն տամ ու ասեմ, okay, հիմա այս visual stimulation-ներն են, հիմա գտեք այսինչ correlation-ները, այսինչ պատասխանների մեջ էլի։ Հա, մերսի։
Խնդրեմ։
Կարելի է Ինձ ուղղակի հետաքրքիր է ձեր կարծիքը, ասենք Neuralink այդ նման պրոյեկտների մասին ու ոնց եք տեսնում դրանց ապագան ու որքանով Հայաստանը կարող է չունեմ մասնագիտական առումով վերապատրաստվել, որպեսզի եթե mainstream դառնա, մենք այդ ինչ-որ թե տարբեր մասնագիտություններից լիստ է, չէ, միայն ինժեներինգ չէ, ու ասենք Հայաստանը ունի պոտենցիալ, եթե դա mainstream դառնա։
Այս պահին կոնկրետ ես ունեմ 2 լաբորատորիա ծանոթ network-ում, որոնք որ ման են գալիս good data scientist-ների։ Ու էդի բան կոչվում է Human Retina project։ Այդ project-ը շատ մեծ է, որովհետև փորձում են համ հիստալոգիայով, համ ֆիզիոլոգիայով database ստեղծեն։ Իսկ database ստեղծելու համար, էլի նույնիսկ վաղվա բանի ժամանակ ես դրան կանդրադառնամ, շատ մեծ քանակությամբ տարբեր data-ների հետ գործ ունես ու պետքա շատ լավ կարողանաս այդ data-ները առանձնացնես, ասենք noise recording-ից կամ find correlations, and you have to develop good apps to do that analysis, and that's where the world is still behind. So we have lots of things that we want to find out, but because technology is still behind what we want to find out, it kind of gets slower. So we get to the threshold that we cannot move anymore forward. And because in Armenia, I can see that data science gets much stronger and the programmers are kind of getting stronger. But the problem is that these labs, they need really reliable connections. So they don't want someone to come and start the job and then say, okay, no, it takes too much time for me, I don't want to do it. So the commitment is very important in that project. That's the thing.
What about biology? So it should be based on this kind of scientific stuff, right?
You don't need to be a biologist to do that. No, there is a data. It doesn't really matter from where zeros and ones comes, yes. So you just get the data sets and you get the order. Like, let's say I want to see this in these data sets and I want you to kind of dissect this information from my data. And for this, you don't need to be a biologist to do that at all. Like, you can get some idea into the project, but you don't need to be a neuroscientist to understand what is happening there. And most of the data sciences are like that. They just don't know. They don't have a clue what is behind scene. Like, how do you do the recordings, how you isolate the tissue, which animals the data comes from. They just get the data and play around with data.
Thank you.
Okay.
Շնորհակալություն շատ հետաքրքիր զեկույցի համար, Հովսեփ Բազարյան, մոլեկուլային կենսաբանության ինստիտուտ։ Ինձ ավելի շատ գենետիկական մասն է հետաքրքրում, ավելի շատ կանխարգելելու համար գենետիկական ռիսկերը ուսումնասիրվել են AMD-ի զարգացման։
Հա, գիտենք մենք գեները, որոնք որ ես կոնկրետ այդ ուղղությամբ գիտությամբ ոնց որ իմ ուղղությունս այդ չի, բայց ես գիտեմ որ զբաղվել են, որտեղ լաբորատորիակար Գերմանիայում, որը որ հենց մեզ կպած էր ու հենց դրանով էր զբաղված։ Ոնց է տեղի ունենում, դու ուղղակի ունես սովորական retinան, healthy retina, and then you have diseased retina, and then you try to get the idea which genes are having the problem, and that's very simple and easy thing to do. And maybe like 20, 30 years ago, it would be very hard question to answer, but nowadays you just have the samples, and there are companies, you just send them, and then they send you the profile, and then you look what is wrong with the genes. The problem is how to fix that. That's the next step that we need to work on, because finding out what genes are not working properly, it's not enough. Because if you kind of try to remove or have stopped this gene expression, something else will come up, some side effects that you need to end up fixing. So that's why we have lots of gene-modified animals, and I did part of my research on them too, by mimicking different modifications in the proteins and the connections, in order to see if this protein is there, if the connections will be stronger or faster or whatever. And yeah, so it's much more longer process to find out how you can fix it, rather than just to see what is the problem.
Okay. Then thank you very much.